Hello, welcome. We are live once again on YouTube. We're also live on Instagram. So if you are joining me on Instagram, um, you want to join up in the comments, you're going to have to come over to YouTube. Otherwise, just hang out on Instagram, enjoy the show. I'm Melanie King. I'm from the Absolute Recap, and I'm super excited that you're here tonight. We have a lot to go over. Probably going to be here for about maybe like an hour and 20. You were at either of my lives last week. There were 2 things that happened in both technical glitches, and I lost my voice about 50 minutes in, so I'm hoping neither of those happen tonight. Can't make any promises, but I ask you to bear with me if that happens to occur. AP Bio Penguins, hello, Mrs. Jones. Raise your hand in the chat if you are an AP Bio Penguin. She is amazing. We have been on Instagram since 2020, doing AP Bio and supporting students. Really excited for Mrs. Jones to be in our chat. You don't know about AP Bio Penguins, oh my goodness, what are you missing? Head on over to her page, ton of free resources, a ton of quizzes, absolutely amazing to get you ready for the exam, which is in 2 days. Absolutely insane. I need to address one of the comments that I've been getting a lot in the past couple of days. It's either a version of, I'm stressed, I'm done, or I'm cooked. And I just wanna tell you, like, if you're um, you you might be cooked a little bit, maybe you're just, like, a little cooked, but you're already doing everything that you can to try and get ready for the exam. So, pick yourself up, just try to stay positive, that's really half of the mindset. You don't have to be perfect to get a 5 on the exam. Now, common myth, it's not a curved exam, but the AP exams are graded on a cut score. I get asked a lot, like, "What score do I need to get a 3?" Or, "What do I need to get a 5?" It varies. No insider scoop, I can promise you. All I can do is take my best guess based on what I have seen, in previous exams or previous scores. Let's aim high. Let's say 85% correct or 85% points to earn a 5. Um, you know, 62% to get a 4 or 68%, and then maybe like 56% to get a 3. I'm making these numbers up, by the way, because they absolutely change every year. Kyle, your 4th AP exam. Oh my goodness, that's so impressive. You all are so impressive. You take such an incredible amount of exams every year. So much that you're working on. But all of it's gonna pay off, and I mean that. Even if you don't remember the information 5 years from now, which you may not, it's the skills that you gain by taking a class like AP Bio, like AP Psych, like AP Lang. Those are going to be the big heavy hitters. All right. The audio is glitching out. I'm actually gonna close one of my tabs here that helped last time. So hopefully that fixes the glitch. But like I said, technical glitches are going to happen. So with that, I want to get us started. It's going to be about an hour and 20 minutes in order to get through all 8 units. So with all 8 units, I want to start by saying welcome. And you can download the study guide that goes along with this live. So if you look at the top of the chat, if you look at the top of the chat, there's a link. You can get your free preview of the Ultimate Review Packet, and there's going to be a link for you to download the study guide that goes with this chat. So I want to just take a quick moment to show you how to do that. If you want to download this, it's absolutely free. And let's pop over to the screen. So that link at the top is going to take you right to this free preview button. And inside the Ultimate Review Packet, there is a lot of resources by unit, a lot of which are free preview, especially in units 1. But I want you guys to focus on the live. So live exam review. And right now we're in unit 5 through 8. So it's this study guide here that I would like you to download. The study guide is annotation. It's going to have some of the copies of the slides that I have tonight and some quick facts for you guys to reference. When you click free preview, it's going to bring you to a place to sign up. You can enter your Gmail or any email really, but you'll see that the cost is $0. So there's absolutely no fee to access these resources that I'm using tonight. Hopefully you find them helpful. And this is just a quick view of what that study guide is going to look like. So it's 9 pages, 9 or 10, I think, this one. And it's going to have learning objectives, some of the slides that I'm going to do, all 8 units, a recap to get you guys all the way through this. It's not going to have everything because we'd be here for hours and hours and hours, but it's going to have a good proportion of the resources that you do need. So a couple more minutes to chat before we start for real. So if you want to pop over and grab that study guide, that is going to be a resource for you. Really appreciate you guys already talking in the chat, but let's be super helpful. There's going to be a couple opportunities in here to do some practice questions, talk through some ideas. So if I'm not able to get to a question because I'm moving through slides, or maybe I miss it because the chat moves too fast, and you happen to know, help each other out, right? We're all here for the same reason, trying to get ready for Thursday's exam. All right. I don't know if you're here because of the podcast, the URP, you happen to find me on YouTube, somebody randomly sent you this link. Either way, the podcast was our first resource. There are 86 episodes that you can use for the podcast. The binging that would take maybe like 4.5 hours if you skipped a few. Obviously on YouTube, I also have a lot of free resources that you guys can watch too. Yeah, Becca, we are all in this together. Thank you for that. That's absolutely true. All right, so let's zoom in. Let's zoom in is our first phrase. It's going to be something I use a lot, actually. Let's zoom in. Yes, there's merch. Had to use my absolute recap mug for us today. I'm going to make myself a little smaller so our slides get a little bigger and we can chat. All right, so units 5 through 8. Heredity is going to be first, really small percentage represented. Then gene expression and regulation. I think universally, unit 6 is everyone's least favorite. 7 is natural selection, and 8 is ecology. If you don't, some people are worried they didn't get to unit 8, or maybe they didn't get to unit 7 and 8. On the other hand, some teachers start with unit 8. Unit 8, in my personal opinion, and all the feedback I get from my students, is the easiest. Because by the time you get there, a lot of your bio knowledge is a little bit more solidified. And then if you've gotten through, lac operons and transcription and translation, ecological niches just seem a little more approachable, and at least macroscopic and observable. So unit 8 is usually everyone's favorite. But unit 5 is where we're going to start. It's not all because a monk started a garden, but it's pretty close. So these are the learning objectives as stated by the CED. I'm not going to go through these one by one, but they are all present for you on your study guide. It's a really great thing to do when you're kind of trying to decompress and go through the information kind of in a broader scope way. So if you are looking to kind of quiz yourself or talk out loud, study with a friend, going through these learning objectives that the College Board has asked you to be able to do is a really good study strategy because it gets you talking and it points out both vocabulary as well as concepts you might not be as confident in. So if you've not seen these, I have them in the study guide for every unit, 1 through 4 as well. Definitely worth referencing. Most important, and I will pause on all of these slides. You don't have to know things about sexual reproductive cycles of plants or animals. Now, some of this information used to be on the AP Bio exam. So if you have a teacher who maybe has been teaching AP Bio for a long time, it is possible they taught you or they went through things with you that are just no longer on the exam. It's not necessarily bad, but these are all things that might be considered illustrative examples. So for what do I mean by that? I mean that if they gave you a genetics problem about fish, for example, they might provide you with information about reproductive cycles within fish for you to apply the knowledge that you do have to know about. And that can be really overwhelming, but we want to kind of like work through all that extra information and get to the real heart of the issues. But let's warm up. All right, I have a diagram on here we should know with A, B, C, and D. Let's type in the chat. Do you know what any parts of these structures are? You don't have to know all four, but if you happen to know one of the four, let's see if we can't collectively figure them all out. What parts do we see here? Oh, some really good terms popping up. Okay, all right. Homologous chromosomes. All right, we're gonna start. We're gonna start here. D, homologous chromosomes. Yes, they are examples of homologous chromosomes, and we know that because they are similar size and similar shape. And if we had more alleles represented, we'd see they have the same genetic information as well. So homologous chromosomes is correct. Let's look and see if we got B. B is a centromere. Great job. So this is going to be where, well, the next one is. Anybody get A? Let me look for it in the chat. C, centromere homologs. I did see it. All right, I see the term sister chromatids. Or sister chromats. Sister chromatids is the answer that we're looking for. More specifically, that's going to reference C. So A is a duplicate chromosome, and this is going to imply that our chromosome has already gone through replication, which would occur during the S phase of the cell cycle. So I have homologous chromosomes. They are both in duplicate form. They are still held to their sister chromatid by the centromere. So C is one example of a sister chromatid. Had I labeled the other arm, it would also be identical, and it would be the sister chromatid to C. Great job. All right, here's the answers. We all got those pretty well in the chat. So when are we going to see chromosomes that look like that? So I'm specifically noticing that that view of chromosomes is one that I might see in metaphase 1. So here we have meiosis. Let's remember, the goal of meiosis is to produce gametes through the process of gametogenesis to create haploid cells. So 2N diploid, N haploid. And we use this algebraic expression because the number of chromosomes that we have within a cell is going to vary from species to species. And there's nothing that says, more evolved species have a specific number of chromosomes. That's just not valid. They're just going to be different based upon how their evolution did occur and how many segments they essentially differentiated into. So when we're talking about meiosis is the process of cell division in order to produce gametes. So gametogenesis is another term for the production of gametes. And the reason why we can say gametogenesis is that can be more specific to a species and specifically that the gender of that species, right? So male gametogenesis in the production of sperm is going to be different than female gametogenesis in the production of eggs because the product will not be the same. So if we look here at meiosis, I can see that my homologous chromosomes are going to pair up. And specifically during prophase 1, I'm going to see crossing over. And that's going to be one of the ways that we are going to increase genetic diversity and ensure that all of those haploid cells that we produce are not only different from each other, but they're also different from the original parent cell. And those new chromosomes are going to be referred to as recombinants. The other term I want to make sure we touch on before we leave meiosis is chromatin with an N versus chromosome, right? So we can see that DNA takes many forms depending upon how coiled and condensed it is. Chromatin would be the loosest form that we see our DNA in, and that's going to be how we find it during interphase, right? When the cell is just doing all of its normal cell stuff, it's not going through any sort of replication. During mitosis and meiosis, DNA must condense into chromosome form for eukaryotes in order to essentially keep track of all of your chromosomes. Commonalities that we find between meiosis and mitosis, they do look really similar, specifically in meiosis 2. Big differences is what is pairing and lining up in metaphase. So during meiosis 1, metaphase 1, we're separating homologous chromosomes. During meiosis 2, we're separating and pulling apart sister chromatids. And so we can see that the actual process of meiosis 1 halves the chromosome number, but all of our chromosomes are still in duplicate form, because if we look at telophase 1, they all still have that X shape. And so my sister chromatids are still attached to the centromere. When we go through all the way in meiosis 2, we finally form our haploid cells. And that's when we get cytokinesis finishing. And if those cells, those gametes needed to continue, the gametes would differentiate into their final shape, right? So I'm seeing a lot of questions about what is gametogenesis. Gametogenesis is the production of gametes, and a gamete is a sex cell, right? A cell that is haploid and has the ability to combine with another gamete through the process of fertilization. So a sperm, an egg, those are both examples of gametes. All right, a lot of information on this slide. And I kind of wanted to brain dump it for you so that you can have it all at once on the study guide. Big concepts that happen in heredity. Mendel's laws, right? So it all happened because a monk started a garden, and Mendel, although not conscious of alleles and not conscious of the process of that genetics worked, he was able to deduce some patterns. And that's really what we take from him, is the patterns that he observed. So the law of independent assortment, and that's basically saying that if you have 6 chromosomes, right, we'll simplify it, there is nothing about chromosome pair number 1 influencing the way chromosome pair number 2 splits, right? If I had a room full of students and I said, please line up in pairs in the middle of the room, and that's the only instructions I gave you, it would be a randomized process. Who was on the left side of the room, who was on the right side of the room, and that's really what independent assortment is saying, is that each chromosome pair, each homologous pair, they're gonna align themselves in any direction to pull apart from that metaphase plate. Second is law of segregation. And this is referring to sister chromatids splitting. So we're going to end up with different genes in different cells because you're going to have them dividing. Good old Punnett squares. Remember that it doesn't matter whether you have maternal or mom's alleles on the left, dad's alleles on the top. What I do encourage you to do is just be consistent with how you list your alleles, meaning if a dominant is present, list it first. And that's just going to help you interpret the phenotypes properly. So physical expression of the trait. When you write a heterozygous, it's not going to be lowercase p, capital P, because it would be more difficult it'd be easier to miss it if you're going through the process that way. Ratios here, genotypic ratio is going to be which alleles do we have, homozygous dominant, heterozygous, and homozygous recessive if you're dealing with regular dominance, or referring to phenotypic ratio, right? The physical expression of the trait. So the classic Mendel example is with the P's and the colors, but we can go significantly beyond that. And a regular dominant recessive is unlikely to be expressed. Other things to watch out for: dihybrid crosses. You don't have to make the full 16-box square to solve those. If you need some shortcuts, I'm going to go over a few in the live review tomorrow. X-linked recessive. Males are more affected in an X-linked recessive disorder because males are hemizygous. They're not heterozygous. They have an X and a Y chromosome. So because of that, if they inherit a recessive allele on that X, and that has the condition, they're going to express it. They don't have an additional chromosome essentially to mask that condition. Other things to watch out for, like blood typing problems, we have codominance in that example, multiple alleles. Multiple alleles means that there's more alleles in a population that affect the trait. You still can only inherit 2 though, right? 1 from mom and 1 from dad. Non-nuclear inheritance is going to refer to traits that are expressed based on DNA not found in the nucleus. So this is going to be things through mitochondria. It could be things through chloroplast, because remember, each of those organelles does have its own genes, its own ribosomes in the endosymbiosis theory, because they were likely their own prokaryotic cells. And so we find maternally inherited components, since only eggs donate and contribute those organelles to the developing offspring. And on the bottom, just a little side note. I'm a Latin nerd. So when we do P generation for the paternal, F1 generation, that's the first generation, the filial generation. F2 would be the second filial generation. It's just the way that they track through the generational concepts of genetics. But we are going to do a quick practice question while I catch up on the chat. And it is color blindness. We're going to practice an X-linked recessive condition. So a woman with normal vision, and she had a dad who was colorblind, and a man with normal vision had children. What is the chance they have a colorblind son? And so that is the question that we are looking for. All right. So let's look here at our SAN answer. I see most people in the chat saying 25%. I have a 0. 100% chance. Okay, so we're all over the place. That's all right. Let's go through. We basically have now said every option. So we have to figure out mom's genotype first. Dad is XY. Dad does not have color blindness, so he must be just XY. But we have to figure out what mom is. And because mom had a colorblind dad, that's shown in the first Punnett square on the left, her dad's across the top. Because she had a colorblind dad, she must have inherited that recessive condition from her dad. She then goes into the second generation and she goes to have children. And as you can see, there is a 25% chance that she could have a colorblind son. 1/4 of that Punnett square box would be expressed. Actually, if the question was what proportion of their children as a whole, and didn't ask about male and female, just asked about chances of having normal vision child, that would be 75%, representing the other 3 boxes. So just an expression to solve the problem when you're working backwards. All right, one more. I once had a student who told me they refused to learn Punnett pedigrees. They said, I will just get that question wrong. Don't be afraid of pedigrees. This is not a great representation because I don't have my generations labeled. I apologize. Normally we'd have generations labeled. We'd have individuals labeled. This is just a blank one. So reviewing some terms while we go through and you guys get your guesses going. We have autosomal as a terminology. So autosomal is going to be referring to a trait found on any of the first 22 chromosomes, like not the sex chromosome. Sex-linked, referring to something on the sex chromosome. And then you have dominant recessive, dominant going to be expressed, whether it is 1 copy of that allele or 2. And then we have mitochondrial showing non-nuclear inheritance. All right. And we have Becca guessing mitochondrial, and absolutely. So let's think about why this is. First off, it can't be a dominant trait. And I know that because I have a female in the second generation being affected when her parents didn't have the trait. So right off the bat, skipping generations, clue, can't be dominant, right? Because we wouldn't see it skipping generations if it was dominant. So that's going to be off the table. Sex-linked, you're going to see a disproportional amount of one sex being affected over the other. I don't see that at all. But dead giveaway for mitochondrial inheritance is because all of the children have the condition when mom has it, right? So I see that happening in 2 generations. On the other hand, I see it being inherited by a son in generation 3 and then passing not to the children. So the question is, why does mitochondrial only pass via the mother? That's a great question. So formation of egg, gametogenesis, meiosis, formation of egg is disproportionate. And what you have is cytoplasm and all the cytoplasmic components being stored within that egg, including all of the organelles. Sperm, on the other hand, as they're designed, are going to only contribute genetics, right? Only contribute the DNA of that sperm to the developing offspring. They don't contribute. Now, this is what the CED says. I will say modern modern data has shown slightly different, but what the CED states is that all of the organelles and the cytoplasm comes from the egg, whereas only the DNA from the sperm is going to be contributed to the offspring. So it is known as a maternal inheritance pattern. So Mother's Day just happened. Go thank your mom for all of those mitochondria. Unit 5 is done. So took about 15 minutes probably or so. And judging by that, we have 45 minutes left to go to get through the rest of the unit. So let's move on to Unit 6, where you are what your proteins say you are. Here are all the learning objectives. As I said previously, I'm not going to go through all of them. It's on your study guide, so make sure you go check it out. If you just joined the chat, you're like, what study guide is she talking about? You can see at the top, link to go get a free preview of the Ultimate Review Packet and all of the resources that I've had for my Unit 1 through 4 live, my Take a Test with Me live I did last week, as well as tonight, and the one we'll do tomorrow. They're all up online. You can grab your copy. Here's what you don't need to know. You don't need to know the names and steps of most enzymes. Okay, there are specific enzymes you are required to know, but most enzymes you do not, for either replication or translation. Please, please, please do not memorize the codon chart. That includes all of the stop codons. It's not necessary. You also don't need to know the details of the biotechnology processes. A big picture is going to be sufficient. Let's start with our cell cycle. So we just talked about chromosome structure, chromatin versus duplicate chromosomes. Everything in gray here, that's interphase. This is just the time when the cell's doing all its cell stuff. This is going to be when the cell is making proteins and replicating and lysing things and communicating, basically how the cell is going to be functioning. After the cell cycle, we're going to go into our cell division phase, mitosis, and making identical copies, and cytokinesis, dividing our cytoplasm. The timing of this, it varies greatly. It depends on the type of cell that you are. And some cells just don't go through with it, right? So G naught, that G sub 0, that's going to be when the cells just vacate the cell cycle. Some stay there permanently and some don't. All right. As promised, I'm losing my voice. I'll pop a cough drop. Let's see if that helps. All right, DNA, anti-parallel, negative charge. Two really big important factors to be mindful of. So here I have drawn to show the inversion of my pentose sugar. Looking for the oxygen of my pentose is a really good clue for recognizing that anti-parallel nature. Remembering that my sugars are going to be labeled 1, 2, 3, 4, and 5, which is why we get directionality identified as 3' to 5'. The fact that the phosphate group has a negative charge is going to be important when it comes down to the way that it interacts through the process of gel electrophoresis and why it has movement for that. Here, the drawing that I have made specifically is showing 2 hydrogen bonds between my nitrogenous bases. So just based on that amount of data alone, we should be able to recognize absolutely that this is adenine, and thymine. Don't worry about the spelling, we'll know what you mean. Adenine and thymine. Great job. Did not draw this one. I'm gonna make it bigger so it's easier to see. So because DNA is anti-parallel, the way that it must replicate is going to be in opposite directions because we can only have synthesis, right, creation of a new strand in the 5' to 3' direction. Be mindful of your verbs, right? You don't want to use pronouns when possible. So I wouldn't say it copies. I would want to say DNA is read 3' to 5' and synthesized by DNA polymerase 5' to 3'. Now, the side that follows helicase, the enzyme that's going to separate our strands, that is continuous. That's the leading strand. The side that moves away from helicase, that is discontinuous synthesis. We can't make it all at once because we don't have enough opened at any given time. And so we end up with these small fragments named Okazaki fragments, thankfully not hyphenated infinitely. It was discovered by a married couple, the Okazakis, so they named it that. RNA primers are going to be involved in this process, so we will have to replace those primers because they will have uracil in them in order to fully create our strand. But the origin site is going to be identified where RNA primers are going to be attached. DNA polymerase will synthesize, and it will continue. The lagging strand will be closed, if you will, and made continuous by the enzyme ligase. Oh, I have to stop. This is amazing. Former AP Bio student, I got a 5. Let's just check each other up. That's amazing. Congratulations. Yes. I've had many a student get a 5, and I think that you guys all can too. All right, let's give it a shot. Here we have a codon chart. Transcribe and translate. So I've given you a sequence of DNA. The goal is to first transcribe into mRNA using our complementary base pair rules, our Chargaff rules. The second would then be to translate. A couple of pitfalls that happen here. Number one, directionality. So I've given it to you 3' to 5'. That means our mRNA transcript should be 5' to 3'. RNA still has directionality too. When you go to read the codon chart, you want to make sure that it's the mRNA transcript that you're putting in, nothing else. Don't put the anticodon sequence or anything like that. And we want to make sure that we are using the codon chart correctly. I'm not saying that you have to transcribe and translate, maybe a piece of that later on in the exam. The other thing that students do incorrectly on this is they write the word stop. So stop is an action. It's not an amino acid. So if we were to have 9 letters long, that's 3 codons. My protein might only have 2 amino acids because one of those codons was a stop. And so you would find that you were 1 less amino acid than codons. So I see a lot of transcription in the chat. We see some translation happening as well. Pop up the answer key in just a second. One other thing to remind you of, transcription occurs before translation. Very easy memory hack for that is the C of transcription occurs before the L of translation. Remember, transcription is DNA into mRNA. You're still in the same nucleic acid language. You're just writing down a shorter message. Translation is moving from mRNA, which are nucleic acids, into protein, which are amino acids. We've totally changed the chemical language. And so we translate to a new chemical language, and that's going to be representative of that process. So let's check our key here. I did not include a stop codon in this example. So we have met methionine to start our polypeptide chain. Remember that methionine, AUG, TAC, big clue here that we are on that pace. A couple things about the codon chart before we move on. The design of it is a piece of evidence of evolution. The fact that we kind of have built-in fail-safes. We can make a mistake and still get the same answer, right? So leucine has 6 different codons, and you still get leucine. So it's often that you can make a change in the third letter and still end up with the same amino acid, but this is a really helpful way to make sure you end up with the same protein, even if there are minor changes. There is a concern at times that the mutation could change the amino acid sequence. And I've seen questions like that on AP exam all the time. Some sort of predict the consequence of X on Y, right? This happens, tell me what happens next. So here it's typically about some sort of error in DNA, asking for the effect on the amino acid sequence, and then the function of something coming up. So since amino acids are all distinct in their design and shape, they also have distinct chemistry, polar and nonpolar. You would get different folding if the swap in amino acids was from a polar to a nonpolar or vice versa. So those are going to be consequences that would affect functioning, right? So if it was a different fold and the function of that protein was reception in a cell membrane, well, then it might not bind with that ligand anymore. So that would be a clue that you had an issue. I'm going to make it bigger so we can see. So replication and transcription, they just have a ton in common. So I thought it would be useful for us to point out some of their overlaps and contrasts here. When you see replication, it's going to be in the S phase of the cell cycle, whereas transcription is going to be G1 and G2. It's when the cell is doing all of its protein, factory stuff. We don't use all of DNA for transcription. We only want to copy the gene we need at the time that we need it. Each side of the DNA molecule, however, can be a template strand for different genes. So it's not just junk on one side. In replication, the process is semi-conservative, right? So half is saved and donated to the next strand because we're reading one half at a time. Well, we're going to copy the whole thing. Nucleotide differences. This is actually unit 1 material that we have between DNA and RNA. It just repeats in this unit. Enzymes. DNA polymerase, RNA polymerase. They synthesize in the same direction. And then we have the difference in cell types, right? So prokaryotes, for replication, one origin site is a circular chromosome, whereas in eukaryotes, you have multiple origin sites and you have multiple linear chromosomes. On the transcription side, we haven't mentioned this yet, but I do think it's important to identify. Prokaryotes, because there is no separation there's no nucleus, right? So there's no separation opportunity between transcription and translation. Everything's happening in the cytoplasm. They happen simultaneously. Literally, the moment that mRNA transcript is produced, ribosomes are going to start translating. But in eukaryotes, we have the nucleus as a barrier. So we'll transcribe in the nucleus. That mRNA transcript is going to go through a processing phase, right? MG-CAT, poly-A tail. We are going to have splicing out of our introns because they are interrupting sequences. And then we will finally get to our translation. From a location perspective, they do both in eukaryotes occur in the nucleus, right? Because there's no nucleus in prokaryotes, we wouldn't see that happening. And lastly, gene expression, before we kind of vacate unit 6. Gene expression is where we get into the land of how do cells control what genes are active at varying times. So what we see happening here is prokaryotes. This is where operons come into effect. Operons can be activated. They can be which are inducible. It can be repressed depending upon what binds to the actual gene itself. RNA polymerase is going to be on that promoter. And because my molecule has directionality, I get terms like upstream and downstream. Eukaryotes, on the other hand, they're going to be influenced by a lot of transcription factors. And sometimes those have to bind jointly in order for that expression to occur. we're going to find that there are also promoters ahead or upstream of where our RNA polymerase transcription factors bind and start. And again, we also have mRNA processing. We're going to take a quick break. And I know that one of our members already talked about other AP exams that they're taking, but take a moment to breathe. We're about halfway through. What AP exams have you taken this week? Do you take this coming few days? Many of us are in the thick of it, taking a lot of classes, have a ton of advice for everyone else. Yeah, I know. Do we know where this quote is from? So how many cups of sugar does it take to get to the moon? It's a quote from a movie from the 90s, cartoon as it was. It's a great movie. What else are we taking? Oh my goodness, a freshman AP Bio is your first one. That is amazing. Good luck. APUSH, AP Calc, BC. Oh my goodness. Wow. AP Lang, Psych, APUSH. Wow, you guys are so impressive. Bio is your last one. All right. Great job. All right, Sarah, we have to address Sarah. Do you think if I pull an all-nighter to study, I'll do better? I don't. But obviously if you want to, but it wouldn't be my advice. And I say that because physically the way your brain has the biology of your brain functioning. So you should do better if you sleep than if you don't. Primarily because things that you put into your brain into short-term memory likely won't be there in the morning anyways. I think that there is a way to reinforce confidence the night before that doesn't have to create a ton of anxiety. And I know that sounds easier said than done, but I definitely wouldn't recommend an all-nighter. All right. Moving on to Unit 7. Population change over time. Some math in both Unit 7 and 8. I'm not going to review math tonight. I will point out where the math is. I'm going to do a little bit of math tomorrow in the live. Learning objectives, volume of learning objectives, not only in natural selection, but also this one. There are no exclusions. You have to know it all. And it's the only unit that doesn't have things that say you don't have to know it. But that's okay. So evidence. There's a ton of evidence. Here's just a quick snapshot of the evidence that they would like you to know. Number one is fossils, right? So not only where they are, but how old they are, where they fit into the scope of the fossil story. Biogeography being where you find them, how we have continents that have shifted over time, how we can talk about the fact that the land has changed, therefore living things have changed alongside with it. We also have structural and functional similarities. So homologous structures, like the arm that I have shown here, versus vestigial structures. And I'm also going to do analogous structures that also trips up students a lot. So when we're looking here at the difference, homologous structures are going to have commonalities because they have common DNA. So the forearm is often used as an example. We look at our arms, one arm bone, two lower arm bones, and a series of fan bones. I might have that bone pattern, and so does a bird. We both share that bone pattern. We have homologous structures because of our DNA. Functionally, they are different, right? Functionally, I'm not flying anytime soon, but that's a homologous structure. Vestigial structures are structures that we keep and maintain because our DNA still tells us to make them, but they don't have a big functional purpose for us anymore. So things like our tailbone, our appendix, we have muscles on our skull that attach to nothing, move nothing, but we still make them because we still have the instructions to make them. It's kind of like leftover DNA. Analogous structures are structures that have the same function, but not the same evolution, right? And I know that because if I were to peel back the layers of those structures and look into the DNA, I don't have a lot in common. So when we look at insect wings, bird wings, they both fly, but we have invertebrates, no bone structure for the insect. We have vertebrate and bone structure for the bird. So that's going to be a big thing that we see here. All right. All right, we're going to take a practice. Experimental evidence shows that the process of glycolysis is present and virtually identical in organisms from all three domains, archaea, bacteria, and eukarya. Which of the following hypotheses could be best supported by this evidence? All right, so let's look at option A. If you know it, just pop it in the chat, but I'm going to talk through the choices while you guys think. All organisms carry out glycolysis in mitochondria. A is not true. Glycolysis occurs in the cytoplasm of cells, so that can't be the choice. B, glycolysis is a universal energy-releasing process and therefore suggests a common ancestor for all forms of life. I like B. Let's look at the other options. Across all three domains, all organisms depend solely on the process of anaerobic respiration for ATP production. Well, solely, no. Anaerobic, that's fermentation, so that's not true. D, the presence of glycolysis is an energy-releasing process in all organisms suggests that convergent evolution occurred. D is a trap, okay? Because we throw a nice buzzword in there, has the word evolution, so why not pick it? So convergent evolution is when you have the same end, but not the same beginning. So organisms have many times developed very similar traits because they have been in very similar environments, experiencing similar selective pressures. And so eyes are a great example of that. And the ability to sense light has happened many, many times over the tree of life, independently developed because similar selective pressures. So the correct answer here would definitely be B as our first step in the oxidation of glucose. So evolution, by definition, right, is the change in allele frequency in a population over time. So we have a lot of terminologies there that we want to make sure we refer to. Alleles being alternate forms of a gene. When we do our genetics problems, that's what we represent as our capital and lowercase letters. Frequencies is going to refer to the part of a whole, either through percentage or fraction. And then a population is also a term that we will have to reference. So a population is going to be a group of the same species in the same geographic area that can interbreed. And that's important because we have to recombine new alleles in order to have that happen and get new phenotypes. So Hardy-Weinberg equilibrium, it's on the equation and formula sheet. Students tend to get tripped up on it. It is a mathematical model for non-evolving populations. So in order for this to be true, there are 5 conditions which have to be met. And they're basically never met in nature. So this is a hypothetical, theoretical, I love, excellent. I'm glad you like it. This is also amazing. Individuals don't evolve, populations do. So true, so true. So first, you have to be really large. Small populations, they don't fare well in the natural world. So really big. Random mating. So there's nothing directionally to be selected for. Everybody is just kind of recombining genes with no pressure. No gene flow. So we're not going to be introducing any new alleles or removing any. And there's no mutations. Like none of this happens in real life. When you are faced with Hardy-Weinberg equilibrium, things to remember. P and Q, by association and by tradition, P is the dominant allele represented. Q is the recessive. If you swapped them, it would be fine. They are fractions of a whole, so they will add up to 1. The way to go about these problems is to go for the recessive first. You're usually given some sort of information about a recessive phenotype, and that's going to be a really good clue for you because that's Q squared. So Q squared, and you can solve for Q. Once you have Q, the whole thing kind of unravels. So that's absolutely my advice. The reason you don't want to approach this problem from the dominant side first is because the heterozygous, that's 2PQ, it gets lost in the math. So go at these types of problems from the recessive angle and you'll be in much, much better shape. All right, phylogeny. Basically how everything's related. So there have been phylogenetic trees in the FRQs before. I've seen it represented for the visual representation question 5, pretty recently actually. The thing about this is when you're making phylogenetic trees, you can only make them based on the evidence that you're given. So they're limited, right? They're only temporary representations. And molecular evidence is by far stronger than any sort of structural or morphological evidence because you're actually going to get to percentage of amino acids in common or specific proteins or specific DNA sequences. Couple pitfalls. Any place you see the branches meet, so the node that I have labeled here, you need to think of this in your brain about that's a swivel point. So currently, it would be incorrect to say that B and C are more closely related because they're nearest each other on the diagram. That's not valid, right? We want to look back to when do they both most recently share a common ancestor. Physical placement isn't going to be enough of a clue because you can pivot at those nodes. Do they share a recent joint on the tree? That's going to be where we go. So let's see if we can interpret some of this right off the bat. I think that's really the best way to go about this, considering the exam's on Thursday. So if we're looking here, I'm going to make it bigger so we can see. We have nucleotide differences amongst 5 species of bacteria. And we want to attempt to say which of the following trees is most consistent with the data. My personal way to go about this is to find the one that's least related first, because that's going to be our out group. And that's going to be really useful in this entire puzzle. So if I look at this data, I can see, first off, all the horizontal slashes, that's comparing this one organism to itself. So 1-to-1 is the same species, and so that's why there's no data. The whole chart isn't filled out because you would just be repeating the information like in a mirrored way. That's why it's half. So the one that's least represented is definitely number 5. Species 5 has 27 to 26 different nucleotides from all the others. So 5 is our outgroup. Both choice C and D are going to show 5 as an outgroup. So then we need to choose between those two. I see a lot of votes for C. This one in particular is amazing. C is absolutely the answer because 3 and 4 are going to share a lot of nucleotides in common with each other, and 1 and 2, well, they only have a difference here of 3 nucleotides. So really, really closely related here, and that's why C is the best answer. I will tell you D looks good because it's nice and neat, doesn't cluster anybody together, but C is absolutely going to be the best answer here. And we're moving into Unit 8. So we're in our final 15 or so minutes here to get through Unit 8, ecology, getting a little bit more Lion King-y, if you will, the circle of life. But Unit 8 is my least favorite unit, I'll be honest with you, but it's okay. Learning objectives, they are on your study guide. You do have to know a lot. And here's why Unit 8 could be really tricky. Now, if you took APES, right? If you have anybody here from APES versus Everybody with Mr. Smeeds, maybe in the past, they will, ecology is a run through of that. So Unit 8 is really, really repetitive of that. The challenge with Unit 8, yeah, I know. I love APE. It's so logical and it's literally APES. I agree. So if you took APES, that will help you. And it is logical, but the challenge to Unit 8 is that because there's no specific examples that you absolutely have to know, it can be a little overwhelming. It can be like, okay, but like now what do I do? Because if I don't technically have to know any of these example ecosystems or any specific organisms or how they interact, just trust that the College Board is going to give you the context for you to know the answer. They're going to give you a lot of background for you to extract the answer from it. And that's really where we want to spend our focus for this section. So, big concept for Unit 8 is energy as well as available biomass. Everybody's competing. They want energy. They want matter. So when we look at trophic levels, troph means feeder, think like the trough of a pig feeds out of. We have our primary, our secondary, our tertiary, our quaternary as you go up the list. Every time you move from one level to the next, tons of energy is not passed because number one, not everybody's eaten. They just they don't pass up the chain. And it's also super inefficient in terms of the transfer of energy. So it's like a 10% transfer. It's really mild. That means the number of organisms or the biomass at those larger other levels, you aren't going to have as many organisms because there's just not enough energy to support many organisms. There's only so much to go around. So typically at the top of an environmental community, you'll have something known as a keystone species. And that keystone species, either through predation or their actions, they serve as like the main puzzle piece in that environment. So if something happens to that ecosystem where like an invasive species comes in or the keystone species has like a infection or goes extinct, the entire community is just absolutely going to collapse. And that's really important to be mindful of. I've seen this in a lot of questions. Diversity is huge for ecosystems. The more species, the better. The greater species richness, the more resilient you're going to find those ecosystems to be because there's more occupied niches, right? So if something were to try to interrupt the ecosystem and the way in which the organisms fill that space, there's just not a lot of slots for them to fill. And so that's generally why better species, more species can be resistant to that. So we already talked about the word population and natural selection, and that's where evolution happens because alleles are recombining. But on the community level is where you have a lot of predation. Everybody's got to get their piece. They're competing for food, water, shelter, mates. Those are the big four. And a lot of the structures, the physical attributes of these organisms and the behavioral attributes, whether it's community structure, whether it's some sort of like parenting structure that you find with certain mammal groups, whether it's going to be like a honeybee hive structure, they all relate back to one of these four things. And so we see that happening over time. Oh, my goodness. Noah, that's amazing. Thank you for that. I appreciate it. We're going to chat. We're going to practice. This is a freebie question. This is from the 2013 AP Bio exam. And like, you don't actually have to know biology to answer it. I'll be honest with you, because it's just telling you that they want you to identify the graph that shows and supports the claim that communities with high diversity are more resistant to change than communities with low diversity. Okay, so more resistant to change if you have high diversity than low diversity. If I look at our two axes here, we have species diversity on x-axis, horizontal, survival of an invader, right? So an invasive species is coming in. They are trying to take everybody down. Are they going to survive more as I go up species diversity, or are they going to survive less? Basically, it has to be C or D. A is flatlined. That's just illogical. And there's no reason for it to bounce back, right? My V that I see in graph B, there's no event happening. So I don't know why it would occur in that way. So I see we're stuck between the two. Let's take a peek at the answer here. And the correct answer here is D, because my invaders, right? My invasive species that's coming into the community, I'm seeing the survival rate decrease of my invaders as I go increasing species diversity. And that's the correlation that we're going to see. if you find graphs like this challenging, and I know many students do because they seem abstract, put numbers on it, right? Just make them up. Give it 5, 10, 15 on both axes and restate and reread the question so that you can actually follow the trend and give examples. So you can pick 2 points on the graph and say, okay, here's 5 invaders that would survive, but here's 2 invaders that would survive. It's gone down, and I can pass that through. All right, population growth models. There are equations. They are on the formula sheet. This is Graph City when it comes down to here. Exponential growth is when you don't have pressure on a population. So if you took AP Psych, this is Malthusian theory, Thomas Malthus, the economist, saying that populations increase exponentially until they are going to receive limited resources. Logistic growth is what we see on the other side. So grow to a point. Once you reach carrying capacity, right, K, carrying capacity, and that's due to space, it's due to resources, at times mates, the population size is going to essentially little mini peaks and valleys. Now, if there is some sort of event, either geological or meteorological or invasive species or predation, hunting, that happens, we would expect the graphs to change again. But logistic growth is just going to be significantly more common than what we see with the others. All right, a few more practice here, and we are almost done. So scientists have found that the existing populations of a certain species of amphibian are small in number, lacking in genetic diversity, and separated from each other by wide areas of dry land. From which of the following human actions is most likely to improve the long-term survival of amphibians? All right. I say we just clone them. Option A, clone the largest individuals to counteract the effects of aggressive predation. Number one, the largest may not be the fittest, right? Fitness, reproductive success, super important. It's not always going to be the largest because then every organism will be getting larger over time. We don't see that happening. So A's out. Reducing the population size by 1/5 to decrease competition for limited resources. Remember, we said small populations, they don't fare well in the natural world. So that's not going to be good. That's also not going to increase genetic diversity. C, make a dam, an irrigation system to control flooding. Did we say it was flooding? Dry land. Okay, areas of dry land. So that's a stretch. D is definitely our best choice because we want to make sure we have the opportunity for offspring to be formed between the two populations. The example that it gave you in the prompt is called geographic isolation, and that's going to cause, over time, speciation. Because if you physically separate, due to usually land masses of some variety, 2 groups of species and you limit their ability to interbreed over enough time, they will no longer be able to interbreed and they will have developed into new species. All right, to recap. So we've got a couple of summary paragraphs here. Let's go big so we can see it. In Unit 5, heredity patterns and probability of alleles distributing during meiosis and recombination during fertilization is important. Remember, genotype influences phenotype, and Punnett squares and pedigrees are going to be asked of you there. In Unit 6, it's all about the central dogma, so DNA to RNA to protein. Know how to use the codon chart, but don't memorize it. Eukaryotes have a lot more gene expression than prokaryotes. All right. Unit 7, this is going to be how evolution occurs, the mechanism, the process. Change in allele frequencies in a population over time. That's going to be your Hardy-Weinberg and the 5 condition. And don't forget about phylogenetic trees. And lastly, looking at ecology. So population and then community leading to the ecosystem. You want to make sure you follow the energy, follow the matter, and you have different levels of competition. And greater diversity is going to be more resilient. So with that, we have one more live tomorrow. So there's actually 2. So I will be on YouTube at 8. It's going to be pretty short, but I do want to leave you with some quick tips and tricks, some last minute survival stuff in order to get you guys ready for the AP Bio exam. If you are an Ultimate Review Packet subscriber and you've purchased the Ultimate Review Packet, I will also be going live in the URP. So the link to that is in the URP. You can grab it there. 8:15, 8:20 is probably when they'll log on to that live, and we'll do a lot of practice. We're going to go through a lot of math, make sure that we're cozy with that in order to get you ready for the final date as well. So I am super excited you guys were able to join me today. The link, right? So if you popped in late and you're just catching up, the link at the top is going to be the place to go to get the free preview. And the live is also in there, just not on the free preview side. So if you were going to join into that as well later on, you are welcome to do so. With that, please tell your friends, right? Like, follow, comment, share. We would appreciate any reviews that you have. I always get, I always get asked for FRQ predictions. Maybe I'll make one tomorrow. Let me see. Let me sleep on it and I'll let you know. You are so welcome. Thank you guys for coming. I really appreciate you. I'm going to preview the chat real quick before we go. Lot of options here. Yes, you are. So here's the trick. 6 FRQs. Every point is going to be worth the same in all of the questions. So just keep going. Just keep going, right? So a lot of students leave the last few FRQs blank, and it might have been the easiest one. So you don't necessarily want to just get stuck on 1 and 2. They're the long ones. They have the most to read. You can go back to those if you want to. If you want to jump to some of the later ones that have maybe a graph that you're really comfortable with or a diagram that you're like, oh, I know what this is. This is no problem. So you can, I promise. I just got to pace yourself through the whole thing. But you guys are super, super welcome. I appreciate you coming to the live tonight. And I hope that you found it helpful. So don't worry if you came late. Totally is recorded. You can go back and watch it later on. Did I take the exam, or are you a grader or a tutor? So I am, I took it in high school, but it has changed a lot since. I will tell you that I am an AP grader. So I will be grading exams, and I have for several years. And I have a lot. I usually grade about 12 to 1,400 exams every summer. So I've seen a lot of that. Are you allowed to skip sections? Do option D first? Yeah, 100%. So you can answer the FRQs in any order as long as you label them. So if you want to go C, D, B, A, no problem. We even will find your answer if you write it on the wrong page, because we have access to your whole book. So that's not a problem whatsoever. All right. The link for the live tomorrow. So we're going to start here on YouTube. And that link is on my regular page. And then the link for the live that happens after that is in the Ultimate Review Packet. And that's going to also be in the folder that says like live reviews. It's the same place where you guys hopefully grabbed your sorry, please give me a 5 if you get mine. If you write something that's wrong, that's right. Well, I get the point. If I get yours, I will try to give. I don't know who's anyone's test, so I can't do that. But write the answer and move on. So if you write something that's correct, and then you write something that's incorrect, it's going to take away that correct point. So you definitely don't want to do that if you don't have to. Yeah, it would take away points. All right. With that, I'm gonna wrap up here. It's 9:10, just over the hour mark. Thank you so much for coming, everyone. And I will see you next recap.